When our cells perform their functions, certain waste products are released into the bloodstream. These are toxic and hence need to be removed from the body. The process of removal of wastes produced in the cells of living organisms is called excretion. The parts involved in excretion form the excretory system. Waste removal is done by the blood capillaries in the kidneys. When the blood reaches the two kidneys, it contains both useful and harmful substances. The useful substances are absorbed back into the blood. The wastes are removed as urine. From the kidneys, the urine goes into the urinary bladder through tube-like ureters. It is stored in the bladder and is passed out through the urinary opening at the end of a muscular tube called the urethra. The kidneys, ureters, bladder, and urethra form the excretory system. An adult human being normally passes about 1 to 1.8 liters of urine in 24 hours, and the urine consists of 95% water, 2.5% urea, and 2.5% other waste products. Reptiles, birds, land snails, and insects excrete nitrogenous wastes as uric acid in the form of pellet or paste with a minimum loss of water and are called uricotelic animals. A survey of the animal kingdom presents a variety of excretory structures. In most invertebrates, these structures are simple tubular forms, whereas vertebrates have complex tubular organs called kidneys. Some of these structures are mentioned here. Protonephridia, or flame cells, are the excretory structures in platyhelminthes, flatworms, for example, planaria, rotifers, some annelids, and the cephalochordate. Protonephridia are primarily concerned with ionic and fluid volume regulation, that is, osmoregulation. Nephridia are the tubular excretory structures of earthworms and other annelids. Nephridia help to remove nitrogenous wastes and maintain a fluid and ionic balance. Malpighian tubules are the excretory structures of most insects, including cockroaches. Malpighian tubules help in the removal of nitrogenous wastes and osmoregulation. Antennal glands, or green glands, perform the excretory function in crustaceans like prawns. In humans, the excretory system consists of a pair of kidneys, one pair of ureters, a urinary bladder, and a urethra. Kidneys are reddish-brown, bean-shaped structures situated between the levels of the last thoracic and third lumbar vertebra, close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measures 10 to 12 centimeters in length, 5 to 7 centimeters in width, 2 to 3 centimeters in thickness, with an average weight of 120 to 170 grams. Towards the center of the inner concave surface of the kidney is a notch called the hilum, through which the ureter, blood vessels, and nerves enter. Inner to the hilum is a broad funnel-shaped space called the renal pelvis, with projections called calyces. Inside the kidney there are two zones, an outer cortex and an inner medulla. The medulla is divided into a few conical masses, medullary pyramids, projecting into the calyces, singular calyx. Each kidney has nearly 1 million complex tubular structures called nephrons, which are the functional units. Each nephron has two parts, the glomerulus and the renal tubule. The glomerulus is a tuft of capillaries formed by the afferent arteriole, a fine branch of the renal artery. Blood from the glomerulus is carried away by an efferent arteriole. The renal tubule begins with a double-walled cup-like structure called Bowman's capsule, which encloses the glomerulus. The glomerulus, along with Bowman's capsule, is called the Malpighian body or renal corpuscle. The tubule continues further to form a highly coiled network proximal convoluted tubule, PCT. A hairpin-shaped Henle's loop is the next part of the tubule, which has a descending and an ascending limb. The ascending limb continues as another highly coiled tubular region called the distal convoluted tubule, DCT. The DCTs of many nephrons open into a straight tube called the collecting duct, many of which converge and open into the renal pelvis through medullary pyramids in the calyces. The Malpighian corpuscle, PCT, and DCT of the nephron are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medulla. In the majority of nephrons, the loop of Henle is too short and extends only very little into the medulla. Such nephrons are called cortical nephrons. In some nephrons, the loop of Henle is very long and runs deep into the medulla. These nephrons are called juxtamedullary nephrons. The efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubule called the peritubular capillaries. A minute vessel of this network runs parallel to Henle's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephrons.